Hello students, this lecture we're going to talk about hearing. This is the last portion of peripheral nervous system afferent, and supposedly we should talk about smell and taste as well, but because this semester we are so tight with time, I decided to eliminate those. That's not like something such fundamental to go over. Definitely we're going to go over hearing. So you'll notice that in this PowerPoint, there are many slides that are hidden. So disregard those slides, okay? We're gonna just talk about the structure of the ear and the physiology of the hearing and the equilibrium as well, which is basically part of the ear. Okay, so first of all, we're going to talk about the structure of the ear. The ear is a receptor organ for hearing and equilibrium, and is composed of three main parts. There is an outer portion, which is indicated over here, a middle portion known as middle ear, and then there is the inner ear, which is basically all this portion. So the outer ear and the middle ear function in hearing. We'll see that the inner ear, besides functioning in hearing, it functions also in equilibrium. So we're going to talk briefly about the three portions, the three regions of the ear. First, we're going to start with the outer or external ear. The external ear is pretty simple. It is actually composed of the auricle or the pinna, which is the one that you can touch very easily, okay? And the external acoustic meatus, which is also known as external auditory canal, which is all this over here, where you put your Q-tip in when you clean your ear. You should always be very careful utilizing Q-tips, not to go too far in, not to ever possibly touch the tympanic membrane. The tympanic membrane is this membrane, which actually is at the very end, as delimitates the boundary between the outer and the middle ear. And it's a very, very delicate membrane. If you damage it, then your hearing could really be impaired. The tympanic membrane is also known as eardrum, and really you can think about it as a membrane, those kind of membranes that are part of the drums. Is a very thin layer of tissue that receives sound vibrations from the outside world and transmits them to the middle ear. And we'll see that in the middle ear there are three very particular ossicles, as we call them, which I'm sure you have heard about already. The tiniest bones of our body are these ossicles of the middle ear. Okay, so the middle ear is composed of the ossicles. We're going to name them. I'm sure that you guys have already heard about these names. The middle ear also contains the eustachian tube, which is also called the internal auditory tube or pharyngotympanic tube here. We're going to talk about it and what it connects to. And the other two elements of the middle ear are the oval and the round windows that we are actually going to talk about quite a bit. Okay, so let's look at the ossicles. The ossicles are very fun. There are these three very particularly shaped bones in the middle ear, known as malleus, incus, and stapes. And they're also commonly known as hammer, anvil, and stirrup. They're more commonly known with these names because they really resemble these tools that we utilize in real life. Hammer, malleus really looks like a hammer. Here it is, okay? and it connects with the incus, which really function and looks like an anvil. And then lastly, there is your stapes over here, which really looks like a stirrup. So these three very tiny bones, are the tiniest bones of the body, are connected with one another, interface with one another, and as you can see, they're connected to the tympanic membrane, and so you can already infer that the vibrations that arrive to the tympanic membrane are transmitted, in fact, to these three bones, and we will see where they end up. Okay, they end up on another membrane basically, which is located on this side. You cannot see it over here. We will talk about that one. Now let's finish with the middle ear. And I told you there is the eustachian tube, okay, which also has different names. Most commonly really is known as eustachian tube. This is a canal that connects the middle ear to the nasopharynx, and its function is to control the pressure within the middle ear, and to actually equalize the pressure of the middle ear with the air pressure outside the body, so that you don't get that weird feeling of having pressure really in your ear. Sometimes this canal can be maybe filled with mucus, and that's when you have these problems where you feel really pressure in your ear. It should not be filled with mucus normally. The other two elements of the middle ear that I want to talk about are the oval window and the round window. Let's start with the oval window. The oval window is this one over here. Is the actual window covered covered with a membrane, kind of similar to the tympanic membrane, on which the stapes ends and connects to. And so when basically what happens is that when the tympanic membrane or eardrum vibrates, the sound waves travel via the hammer and anvil to the stirrup or stapes, and then in turn there is a vibration of the oval of the membrane on the oval window. Okay, and we'll see how these vibrations then keep traveling in this other structure that you see here, the cochlea, which we're going to talk about in a minute, which is part of the inner ear. Now, the round window is the window down here. This round window is also covered with a membrane, and basically this is where the vibrations end up to. The vibration coming through the oval window, and they pretty much end on the round window. And the outward movement of the round window, the membrane of the round window, allows the fluid to move within the cochlea, so to kind of keep moving, and thereby making the activation of the auditory receptors possible, as we will see. All right, now we're going to talk about the inner or internal ear. This is very interesting. This is where everything really happens in terms of translation of mechanical vibrations into nerve impulse, which are going to travel through, of course, the auditory nerve through your brain. So the inner ear, in the inner ear, we can recognize two areas. Here highlighted in gray and purple. These two areas are the bony and the membranous labyrinth. Why labyrinth? Really is a labyrinth here. And each of these two regions contain structures that we're going to talk about. The bony labyrinth, again in gray here, contains the cochlea, the vestibule, and the semicircular canals, okay, which are these three structures kind of oriented as that you would orient the axis to X, Y, and Z axis in space. And then we have the membranous labyrinth, which contains the cochlear duct, which is um, this purple structure that follows the cochlea. Inside is a duct, okay? And also the utricle and saccule, which I will see these enlargements over here, respectively. The saccule is this one, and the utricle here. These contain very important elements for equilibrium. And the semicircular ducts, which are these purple ducts inside the semicircular canal. Okay, so we're going to talk about all these structures one by one. Now, the one thing I want to make sure is that you don't get disconnected here. When you study the ear and all this formation, you always have to keep in mind that this structure is meant to acquire vibrations from the outside world and translate them into nerve impulse. So don't ever forget that there is a connection to the nervous system here. And in fact, as we will see, the cochlear nerve, which is the acoustic nerve, really connects with those very elements that sense, that those cells that sense the vibrations and are able to translate the mechanical vibrations into nerve impulse, basically action potentials, okay? And therefore, you would expect that they connect with actually the axons of nerve cells. And in fact, that's exactly what happens. And there is also, so there is the cochlear nerve and there is the vestibular nerve. The vestibular nerve is more dedicated to equilibrium, sense of equilibrium, where the cochlear nerve, which really is your acoustic nerve, dedicated to hearing. Okay, and then there are ganglia that we can recognize over here, superior and inferior. They're part of basically the vestibular nerve. Okay, so don't forget about this aspect while we talk about the structure and physiology of the ear and hearing. Now, I want to start introducing you to the physiology of hearing. We have talked about the structures now, okay? And I have to point out a few other names and say substructures that we can recognize, especially in the cochlea. Talk about the cochlea, this spiral-shaped structure. Now, when we take a section of the cochlea, we can recognize two portions. One is superior, one is inferior, and then there is the canal in between. The superior portion here is called the scala vestibuli, this one, and the inferior is called the scala tympani. So scala means ladder, basically, so that's why there's it's kind of a ladder too. You can think about these two structures as ladders, and in here in between, to delimitate the scala vestibuli and the canal, there is the vestibular membrane, whereas the membrane in between the canal and the scala tympani is known as basilar membrane. And in between the scala vestibuli and the scala tympani, there is the scala media, which actually is the one that contains the canal. Okay, so now let's consider what happens when sound waves hit the tympanic membrane. Tympanic membrane vibrates, and it of course transmits the vibration to the ossicles, and eventually to the vibrations arrive to the oval window. From the oval window, pressure waves begin to travel through the scala vestibuli, as shown here, and all of these mechanical waves, sound waves, displace a region of the basilar membrane, as shown here, that contains very special cells. We will talk about this in detail. And special cells that have hair. So remember when we have talked about projections, cell projections, these are, oops, this is a cell. A cell can have at one surface can present these projections, okay, this hair, okay, and that's what these cells look like, okay. So hair cells in this spiral organ detect the movement and are able to translate the mechanical vibrations into release of a neurotransmitter that then depolarizes neurons that are in contact with them, giving a nerve impulse. This nerve impulse will then travels to the cochlear nerve of the auditorial nerve, and the remaining pressure wave vibrations will actually travel back here to the scala tympani and exit the inner ear via the round window that we talked about over here. So let's talk about these special cells that are present here, the basilar membrane that are responsible actually to translate the mechanical sound vibration into a neural impulse, basically. These cells are contained in a specific area known as the organ of Corti, from the name the Italian scientist actually that studied and described this structure for the first time. It's really an organ, okay? So we call it organ of Corti, which is, again, so it's nothing else than a specialized epithelial cells that have this hair shown over here, okay? And but they are not strictly epithelial cells, because they are able to basically release neurotransmitter to nerve endings here of the nerve processes of the auditory nerve cells, okay, and so that they can depolarize those cells and elicit action potentials. And so the organ Corti allows ultimately for the transduction of sound vibration to neural signals, okay? Remember that. Allow for the transduction of sound vibrations into neural signals, action potentials in the auditory nerve cells. And the organ of Corti is located on the basilar membrane and contains these specialized hair cells. Really, there are different types of hair cells. The inner and the outer. The inner hair cells are the ones that transduce the sound vibrations into neural signals. And really, the hair, that are what we call hair, in reality, we should call them cilia. Remember from cells? When the study of cells that we studied this particular projections of the plasma membrane, really, of cells that can be many times only on one side of cells. They're known as cilia, okay? So these are known as stereo cilia, okay? They're the hair, what we commonly call hair, on these very special sensory cells of the organ of Corti. And above the here, kind of in contact with the stereocilia of these hair cells, there is this membrane, is known as tectorial membrane, has this very particular shape, which can move in response to pressure variations in the fluid-filled tympanic and vestibular canals. Now, I want to point out that all these canals that we, generally speaking, the canal that we talked about, which is subdivided into vestibular canal, tympanic canal, and cochlear duct are all filled with a fluid is known as lymph, okay? And depending on where we found the lymph, this can be perilymph, endolymph, but don't worry about remember all this. Just remember that it was pointed out in one of my previous slides that the labyrinth here is filled with fluid called lymph everywhere, okay? So here in this representation, we are sectioning, here we're sectioning the cochlea, okay? This is filled, the scala vestibuli and the scala tympani and scala media, they're all filled with fluid, okay? There is no empty space really. Everything is filled with fluid. And then when we go to zoom into this structure over here, the scala media, that's also filled with fluid. Okay, so all right, so with that said, I just wanted to show you an idea of how things happen here. Sound waves are transferred to the fluid in the cochlea and then to the basilar membrane, which contains the organ of Corti. And regarding the auditory pathway from the organ of Corti, remember that okay, these cells, the specialized auditorial cells in the organ of Corti, they are in contact with the processes here of nerve cells that form altogether, convey to form the cochlear or acoustic nerve. And this travels to the medulla oblongata and to the thalamus, and then to the temporal lobe of the cerebral cortex, which is to the temporal lobe, which contains the cerebral cortex, which is your primary auditory cortex over here. Okay, it is the pathway. Okay, and so that's the auditory pathway from the organ of Corti. And in the next slide, I have very interesting, some very interesting, useful videos to really show you how the process of hearing happens. Let's see if we can play at least one of these videos. First one.
Have you ever wondered how sounds make their way from the source all the way to your brain? Take a trumpet, for instance. When it's played, it makes sound waves in the air. The outer ear catches the waves, which then travel through a narrow passageway called the ear canal. The sound waves reach the eardrum, which is a membrane roughly half the size of a dime. They make the eardrum vibrate, which in turn vibrates 3 tiny bones called the malleus, incus, and stapes. These bones amplify or increase the sound vibrations and send them to the cochlea. The cochlea is shaped like a snail and is the size of a garden pea. It is filled with fluid, and the sound vibrations make this fluid ripple, which creates waves. Hair-like structures called stereocilia sit on top of hair cells and are grouped together as hair cell bundles inside the cochlea. The hair cells inside the cochlea ride these waves, and the hair bundles are moved. The hair bundle on top of the hair cell turns these movements into electrical signals. As the hair bundles are moved, ions rush into the top of the hair cells, causing the release of chemicals at the bottom of the hair cells. The chemicals bind to the auditory nerve cells and create an electrical signal, which travels along the auditory nerve to the brain. Different hair cells respond to different frequencies of sound. The hair cells at the base of the cochlea detect higher-pitched sounds, such as a piccolo or flute. The hair cells toward the top of the spiral detect progressively lower-pitched sounds, such as a trumpet or trombone. At the very top or apex of the spiral, the hair cells detect the lowest pitched sounds, such as a tuba. The auditory nerve carries the electrical signal to the brain, which interprets the messages as sounds that we recognize and understand.
All right, so that's a very interesting. There are many more videos that I'm suggesting you to watch if you cannot understand, especially if you're having problems understanding the process and physiology of hearing. I strongly suggest you to watch the other videos. Okay. Oops. All right, so talking about the equilibrium structure and pathway, this is composed by the vestibule and the semicircular canals. So let's look at these two structures in detail. The vestibule contains two regions known as the utricle and the saccule, which house the macula. When we look at the macula, and here are the utricular and the saccular macula, there are spots basically where special cells are located. Now, if you look at these special cells, you will recall the shape of the cells that we have seen in the organ of Corti. These are pretty much very similar cells. They have these stereocilia, okay, that are located on one of the surfaces of the cell, the apical surface. And these are the cells that actually initiate these are the cells that initiate action potentials in the branches here of the vestibular nerve cells. And so looking at the macula, which is this structure here, we find these hair cells, as I told you, which has the serocilia. Now, these are in contact with a membrane known as statoconic membrane, which has a gelatinous layer containing special little crystals of calcium carbonate known as otoliths. And basically, these otoliths mean ear stones. They are little stones, and these are very important for the transmission of. Remember, this is an equilibrium organ, so for sensing the equilibrium. And so so what happens is that when the head is held erect, the macula in utricles is oriented in the horizontal plane, shown here, and whereas in the saccule is oriented vertically, and the gravity pulls on the otoliths, which because of the gravity, they will deform the gelatinous mass and press consequently on the stereocilia, of the hair cells. And this is pretty much a constant force that is applied, and therefore the hair cells in the utricle and saccule provide a tonic information, which means a constant pressure, a constant signaling, which gives information about the orientation of the head. So the one thing that is important here to point out is that the two maculae, the macula of the saccule and the utricle, they're pretty much both have the function to sense equilibrium, but the macula of the saccule function is to detect vertical linear acceleration, whereas the macula of the utricle, because is disposed in a horizontal position, detects horizontal acceleration. So they are the same function, but determine two different types of acceleration. And they both pretty much function the same exact way via the otoliths that sense the gravity and deform the gelatinous mass of the otolithic membrane that touches them constantly, basically. All right, so this was the vestibule, and now we're going to look at the semicircular canals. The semicircular canals contains semicircular ducts and ampullae. This is an ampulla inside the duct, and there are several ampullas, and basically the mechanism here is very similar. Again, you have these hair cells, okay, located in the crista ampullaris, which is really the organ here, the function in again in maintaining equilibrium, but this organ is more specialized in sensing. It's more of a sensory organ of rotation. Basically, when you rotate your head, this organ is what is really senses the rotation and is activated. So there are several pairs, precisely 3 pairs of cristae ampullaris, which are these structures that are shown here, which function pretty much this very similar way that we have just described. Okay, so we can say that the function of the crista ampullaris is to sense angular acceleration and deceleration as well. So it's another little organ of equilibrium on its own. Okay, so I just wanted to show you now, sorry, this was another slide here, to show you just pretty much another picture here, another detail of the cristae ampullaris, and there are 3 pairs, okay, 1, 2, and 3, and with the hair cells. I don't know if you can see them very well down here. Now, talking about the equilibrium pathway, okay, remember that ultimately there is a nerve there. This is the vestibular branch of the vestibulocochlear nerve. This is eventually the convolutes in the same nerve with the other branch that is the cochlear one. And you see how all these that nerve processes, the cell processes, take contact with all the hair cells of all the little organs of equilibrium that we just talked about. Okay, and so there is a transmission of signal. Of course, there's going to be action potentials that fire here in this nerve, and the information, this equilibrium pathway specifically, of course, transmits information on the position and movement of the head, but most information actually goes to the lower brain centers, which are reflex centers. Is not much of a conscious response that you have. This is more non-voluntary subconscious response, and the position of your head is maintained, especially when you were awake, just maintain upright, straight, even without you thinking about it. Okay, and so it makes sense that most of the information travels to the lower brain centers, the medulla, also some to the cerebellum. Remember, the cerebellum is involved, in fact, in equilibrium, maintaining of the equilibrium. We actually talked about this. All right, so pretty much going to stop here. There are other slides after this one, but As I told you, from this point on, for the olfaction, all the olfaction and gustation material, just disregard it. Okay, we're gonna close the book here. This semester has been a little bit particular, so we're gonna stop here. We're not gonna include that those pathways for our course. Okay, so make sure you know the structure of the ear. This is not very difficult, and especially when it comes to the middle ear, the three little bones. I'm pretty sure that some of you remember them from school, studying sometimes, or they're very, very commonly known. And mechanism, the physiology of hearing. Okay, the organ of Corti is extremely important. So know the organ of Corti and the hair cells with their these cilia, stereocilia, that are responsible really for basically are the key cells responsible for hearing. Okay, they are able to translate the mechanic vibrations into action potentials that are fired in the nerve cells of the auditory nerve processes of the auditory nerve cells. Okay, and the pathway is important. And then we talked about watch more movies. We watched this one, the first one, watch all of them if necessary. They're all very good movies. And then we just talked about the equilibrium, the vestibule, and the semicircular canals. Remember that the types of cells also for equilibrium are pretty much hair cells, the same pretty much cells that we have seen for the organ of Corti. Okay, the stimulation is slightly different, but pretty much the type of cell is the same type of cell. All right, so I'm going to stop here.